Hello. Today we're going to be talking about action potentials. This comes from chapter 7 of your Powers Exercise Physiology textbook. We're going to look at what action potentials are and how they're generated. By the end of this short video, you should be able to answer the following questions. How do nerves generate action potentials? What parts of the nervous system are involved in muscle contraction? What causes resting membrane potential, depolarization, action potential, and repolarization? And how is an action potential transferred from one cell to another? At the end of the lesson, if you really want to test it, if you know your stuff, see if you can describe this diagram over here. This is a diagram of an action potential, and see if you can describe all the events that happen during this process. So first of all, what parts of the nervous system are involved in a muscle contraction? It really requires a lot of your body. A muscle contraction starts in your brain. You start to think about it, and different parts of your brain send information to what's called the motor cortex. And they send action potentials themselves, which we'll learn about in a second, to the motor cortex. And if there's enough information sent there, enough action potentials that reach the motor cortex, it will decide, hey, we need to send this on down to the muscle. So that action potential will travel down the spinal cord, converge onto a motor neuron, and that motor neuron will then synapse with the muscle and cause muscle contraction. Right? So you have the action potential generated first up in the brain, and then it travels down the spinal cord to the motor neuron. The motor neuron then transfers it to the muscle, causing contraction. So an action potential, it's electrical activity, it's voltage. Where does this come from? Right? Most of the electrical activity that we're talking about deals with ions, and it deals with the movement of ions across the cell membrane. Two of those most important ions are going to be potassium, represented by K, and sodium. Now in your cells, especially your neurons, you generally have a lot of potassium inside of the cell and not much out, whereas you have a lot of sodium outside of the cell but not much in. You have these natural, or these gradients. They're not so natural. We'll talk about that in a second. So really, your body makes these action potentials and these voltages by having different concentrations of positive and negative molecules inside and outside of a membrane. And that's what we illustrate here. This is an action potential. This shows at rest the membrane voltage. The voltage of your neuron is somewhere around -70 millivolts, and then you get some inputs into it to either get it closer to what's known as the threshold or further away from threshold. If you want an action potential generated and you want like a muscle contraction, you want to get close to threshold. If you want to not have a contraction, you go further from threshold. Once you hit the threshold, then a lot of positive ions flow into the cell, generating this high spike, which then the ions flow back out of the cell and everything gets restored back down to the resting membrane potential. So really the action potential is this transient spike in membrane voltage where it flips from being negatively charged to positively charged. And I'm going to give you a little bit of an illustration to show you how this happens. We're going to walk step by step through this action potential diagram and tell you what's going on inside and outside of the membrane. So this gray bar right here represents the cell membrane. And on the left side, this would be the intracellular fluid. On the outside would be the extracellular fluid. On the inside of your cells, you have a lot of negatively charged proteins and phosphates and other things. They're stuck in there. They can't go out of the cell, and they have a negative charge. So that contributes to why we have a resting voltage of -70 millivolts. Another reason we have a negative charge at rest is something called the sodium-potassium ATPase pump. Now this pump requires ATP, as the name would indicate, to put 3 sodium ions out of the cell in exchange for 2 potassium ions into the cell. Now look, they're both positive, right? Sodium ions and potassium ions, they're both positive, but what we're doing is we're kicking 3 positive charges out of the cell in exchange for 2 positive charges. So it's a net of 3 going out, or 3 go out, 2 come in, so it's a net of we're losing 1 positive charge, or we get increasingly negative with this. Now the reason it requires ATP is, if you remember, we have a lot of sodium outside of the cell already. And so to get that sodium out of there, you need ATP or energy. It requires effort, energy, to put it out here because it's going against a gradient. Naturally, that sodium would love to go from an area of high concentration to low, but that's not what we want. We want the sodium to go store it up on the outside of the cell, and so that's why it's an ATPase. It requires ATP to do that. So we have what's causing the resting membrane potential to be negative. We have negative proteins stuck inside of the cell. We have the sodium-potassium pump, and we also have other ion channels that result in a negative charge. We have these sodium channels. Now, this is my wonderful illustration of this. You have sodium piling up, and you can see there's this big gradient over here, right? Partially because we're pumping a lot of the sodium out of the cell. And so we have a lot of sodium building up, a high gradient, but that sodium doesn't really like to go against that gradient. This channel, these ion channels, don't allow the sodium to go through very well at rest. Whereas the potassium channels on the inside of the cell, you can see we have a potassium gradient up here. They're leaky, and they allow that potassium to go out. And so some of the potassium that builds up on the inside of the cell will leak out, so we're losing positive charge that way. Right? So the positive charges don't come into the cell at rest, but they do go out of the cell from these channels. And so that's another reason why you have a negative resting membrane potential. And so that's why we start off with a negative membrane potential, about 70 millivolts in a neuron. The next step is going to look at how we get closer to threshold. And this text just summarizes what's going on here. You can read it and pause if you want to read that. The next step has to do with getting the membrane potential, or the voltage, up to what we call threshold, which is this dotted line here. And what threshold represents is actually the voltage at which this sodium channel will open up and allow the sodium to flow into the cell. This little cork or stopper here, it's sensitive to voltage. And once you get a voltage around -55, then it opens up and all that sodium will flow in. So how do we get up there? Well, you have on your neurons, right? So we're looking at a postsynaptic neuron right here. This neuron is receiving information from two different neurons, other neurons. An excitatory neuron in green and an inhibitory neuron in red. That excitatory neuron, when it'll release things like acetylcholine, and that acetylcholine will bind onto the dendrites of the neuron and cause a little bit of sodium or other positively charged ions to flow into this inside of the cell. These are sometimes called graded potentials. They're just tiny little stimulations. One neuron really isn't enough to get enough sodium in the cell to get it to threshold. So look, we have multiple stimulations here, right? So in this first little grade right here, we get a small stimulation that results in some positive ions flowing into the cell, but not enough to get it to threshold. And then we get going down again because we have an inhibitory neuron saying, no, we don't want to generate an action potential, and it actually causes negative things to go into the cell or positive things to go out of the cell. The effect is that we're getting further away from threshold. Once we get enough positive potentials here, excitatory potentials, we'll get up to threshold, right? We get up to this voltage, and that cork will open up. So we have positive things coming in. Once you get enough positive things coming in, the cork will open up and cause a depolarization. And all of those sodium ions that are built into the cell, or that are on the outside of the cell, will flow into the inside of the cell. And that's why you get this rapid depolarization, or going from negative to positive depolarization. That's all caused by sodium ions flooding into the inside of the cell. And the reason they flood in is because you had that cork open up on the voltage sensitive sodium channel to allow the sodium to go down the gradient inside of the cell. All right, so that's what gives the spike in the action potential. But now how do we get back down? All right, so we're at this point we're at the top of this peak here. We have a really high potential. And the reason is because look where all of our positive ions are now. In the beginning, we had sodium on the outside of the cell and potassium on the inside. Because we opened up that gated channel, now we have potassium on the inside and sodium on the inside. Two positively charged ions, they're all on the inside of the cell, so that's why we're very positive. So how do we get negative? Well, the next thing that happens is actually these potassium channels, they'll open up. They're also voltage sensitive. And so when you hit a voltage, this high voltage up here, they open up and allow the potassium now to flow out of the cell. And so that's where the voltage starts to get a little more negative. Now we don't have both sodium and potassium on the inside of the cell. Some's inside and some's outside. But look at what's going on here. We started off in the beginning and we had the potassium on the inside of the cell and the sodium on the outside. But now we're all mixed up, right? We have the sodium's on the inside and the potassium's on the outside. That's not where we want it. And so how do we get back up to where we were before? Well, we do that with the sodium-potassium ATPase pump. It works to restore the gradient. It puts everything back into order, and it's going to work hard using ATP to get the potassium back on the inside of the cell and the sodium back on the outside. And that's how that gets restored to a resting membrane potential. Now, one last thing about this, you might notice right here that this spike, after the action potential, the spike can go down below resting membrane potential. And that spike is actually called hyperpolarization. And that happens for a variety of reasons, but really the net effect here is that when you're hyperpolarized, you've had so many of your potassium ions flow out of the cell that you get even more negative than normal. You're further from threshold than normal. So at this point, when you're in that hyperpolarized point, it's going to be harder to generate an action potential. And so that means if you're in this hyperpolarized point, also called a refractory period, it's going to be harder to generate an action potential and going to be harder to cause a muscle contraction, or at least a twitch there. So it's kind of an off time, or a time where you're not going to be able to generate things as easily. Okay, so that's how we get the action potentials generated. That's what's going on at each point in that diagram. So make sure you can explain that. So once you have that, one other thing that I should add in, these sodium channels, once you have the sodium flow into the cell, let me move back. So once we have this sodium flow in for that action potential right here, we've generated that action potential. The next sodium channel down the line, so we have a sodium channel here, there's probably another one somewhere down below this potassium channel. It will sense that flow of sodium in, that change in voltage, and that will open up. And that will allow more sodium to come in, and then the next will open up, and the next will open up, right? So it's just like dominoes flowing down. You get this one to allow some positive ions in, causes a positive potential. The next sodium channel senses that nearby disturbance. It allows stuff to flow in, causing the next one to sense a disturbance, and that's what's going on. So now that's how it's transmitted all the way down an axon. Now once you get to the end of the axon, what happens is the end of the axon has what's called a Bouton, or a synapse at the synaptic cleft, and that Bouton, or the end of the axon, will release neurotransmitters. And those neurotransmitters will go out from the end of the axon and bind onto receptors on the next cell. Now if the next cell is a neuron, then it binds onto a neuron, or if the next cell is a muscle, it'll bind onto the muscle. But those receptors on the next cell will receive that neurotransmitter and then do that whole graded response again. If it's excitatory signal being released from the end of the axon, it'll get closer to threshold. If it's inhibitory, it'll get further away from threshold. And so that's how it's transferred from one cell to another. And so then you see what happens with the muscle. We've already talked about that. Feel free to look through that in the PowerPoints. Take time to review. Make sure you can explain what goes on at each point in this diagram. That's something that is very important to the rest of what we'll talk about in exercise physiology. So take time to review that in your own studying. And again, these are the main outcomes that you should be able to answer by the end of today. If you can't, go back and look through the PowerPoints and you should figure it out. All right, that's it for today. Thanks.